Hello students, today we will see how bacteria can be identified with the help of biochemical test. So one of the most important tests for identification of bacteria is IMVIiC test. So IMVIiC tests are a group of individual tests using in microbiology lab testing to identify an organism in the coliform group. A coliform is a gram-negative aerobic or facultative anaerobic rod which produces gas from lactose within 48 hours. The presence of some coliform indicates facial contamination. So the term IMVIiC is a acronym for each of these tests. I is for Indole test, M is for Methyl Red test, V is for Fox-Proskauer's test, and C is for citrate test. The lowercase I is merely for in, as the citrate tests require coliform samples to be placed in citrate. So these tests are useful in distinguishing members of the family Enterobacteriaceae. So this IMVIiC test is generally used for identification of bacteria. Indole test. The first one is Indole test. It is performed on sulfide indole motility SIM, shortly it is called as SIM media, or in tryptophan broth, or in motility urease indole medium, shortly known as MIO medium. Result is read after adding Kovacs reagent. The positive result is indicated by a red layer at the top of the tube after the addition of Kovacs reagent. The negative result is indicated by the neck of color sands at the top of the tube after the addition of Kovacs reagent. Next one is Methyl Red test and Fox-Proskauer test, and both are done in Methyl Red Fox-Proskauer broth, but the reagent that are added varies according to the test. So in Methyl Red test, positive Methyl Red test is indicated by the development of red color after the addition of Methyl Red reagent. A negative Methyl Red test is indicated by no color change after the addition of Methyl Red reagent. In Fox-Proskauer test, negative test is indicated by a lack of color change after the addition of Bridge A and Bridge B reagent. A positive Fox-Proskauer test is indicated by the development of red-brown color after the addition of Bridge A and Bridge B reagents. In citrate utilization test, the test is performed on Simon's citrate agar. Negative citrate utilization test is indicated by the lack of growth and color change in the tube. The positive citrate test result as indicated by growth of a blue color changes.